Good afternoon, everybody. Nice to see all of you. Okay, thank you for coming. My name is Dr. Raj Gopalan. I'm the Chief Medical Information Officer and Global Head of Clinical Decision Support for Siemens Healthineers Diagnostics. So I'm excited to share about this topic of AI in lab diagnostics, especially in terms of how we can use AI for ordering the appropriate testing and also how to interpret the results. So glad you're here. And there are going to be two presenters. I'm going to present the first 20 to 25 minutes or so. And then Dr. Wheeler, who is the assistant professor and also the medical director for University of Pittsburgh Medical Center. So she will be presenting following my presentation. And then we will definitely have a lot of time for questioning. So let's get rolling. This is a picture of a brain-to-brain loop concept. This is first proposed by Dr. Charles Lindbergh from Harvard University. So the interesting thing about this is because I usually like to move around the stage, but because I'm kind of stuck here, so I'll try to do my best. See, in this circle, as you can see on the top side, there is a patient's, the physician's brain, right? And at the end of the loop, there is a patient's brain. So in brain-to-brain loop, you can divide that into two parts. One is the manual process, and the other one is the automated process. So as you know, in the recent times, a number of things have been automated. Like, for example, the moment the blood is drawn from the patient, it is labeled, it is transported to the laboratory, and then it is verified whether it is, whether it is not lysed or anything like that, ready to be analyzed. And then it is validated, and then the results are reported back. So all this process is pretty much automated because of cutting-edge information technology, highly automated robotics, and also highly advanced analyzers. So those things are very automated, and it is very, very predictable. So it is very consistent across different labs, and the results are pretty consistent as well. But if you look at the beginning of this loop, so let me walk through the process. So I am an internist, general GP or general practitioner. So when a patient walks into my office, I take the history, do physical examination, and then after that, I kind of within my head configure a bunch of diagnoses. So what potentially the patient may be having. So based on that, I have to order the appropriate test. So what typically happens in the US, as you know, they order panels of tests. And especially in Europe and in Asia, they usually order only limited tests, or the tests that are absolutely required. So even in the US, there is a new effort called Choosing Wisely, because the payment system has changed, and you cannot order and get expected to get paid for all those orders that you make. So you got to be extremely careful in what exactly you would want to order. So you really want to order the tests that are appropriate, that are going to give you more information to be able to rule in and rule out the diagnosis. So that is extremely important. The interesting thing about this is that there are about 14 billion tests available, that is being ordered like every year, okay? And there are about 4,000 different types of tests that you can order. So the amount of tests has kind of exploded. And if you look at the medical school curriculum, when I went to medical school, I hardly spent about 15 hours learning about lab diagnostics, right? So in a 5-year curriculum. So imagine my knowledge in terms of what exactly I should be ordering, and how to interpret the test results. So what happens as a result of that is a number of mistakes are being made. It has been reported that 20% of the time, the physicians make wrong diagnosis. So the impact being that it is going to unnecessarily, make the patient stay a longer time, go through unnecessary, additional testing or procedures, and it is going to increase the cost of care. So, and ultimately, it might even impact the outcome, right? So a number of malpractice lawsuits have been filed when this happens. So there is a potential for lab diagnostics. So it's kind of interesting because nowadays, with the panels and things like that, there are about 40 to 50 different tests that are being ordered. And human brain cannot process, it's not capable of metacognition. So basically what happens is intuition can only scale to a certain extent, but going through multiple tests is a multidimensional problem that the humans are not capable of doing. But the computer is really very good at that. So imagine you have a bunch of tests, 40 to 50, and what the lab currently does is that they actually report the results with reference intervals, right? So typically, if you ask the physicians, and I have talked to a number of physicians, how do you kind of review the results? They say, okay, we'll the results and literally go through the flags. If there is any high or critical values or low critical values, we'll just go through it. And even there is a high, and if the test results are kind of just over the limit, yeah, it's kind of normal, we just move on. But what do you think about that? If you think through it, the reference interval itself is, I learned about it recently. Do you know how the reference intervals came into being? It is basically about 50 years ago, they came up with the reference intervals. Before that, it was not there. So they asked volunteers, about 100 of them, just 100 of the self-declared healthy volunteers. So who do you think they are? Who can, at lab, where can they access the volunteers? Lab technicians and people who are working in the lab. So they give a questionnaire and ask them to fill it up and saying that, okay, if you're normal, then we're going to take the blood, right? So they take the 100 volunteers and then plot it in a Gaussian distribution and then take 2 standard deviation, which is 95%, as the reference interval. So if you think about it, originally it was called actually normal intervals, but people said, what do you think, 100 individuals self-declared, and you're going to call that normal? Then they changed it to reference interval. It's just based on a reference based on the 100 volunteers. And the interesting thing, which a lot of people don't know, is that it doesn't include anybody over 60 years, right? I mean, labs, people won't be working there over 60 years, so it doesn't include people over 60 years. So the reference intervals are actually not valid for people over 60 years. And you can imagine most of the conditions are more common in older individuals. So this is pretty mind-boggling if you think about it. And if you have a bunch of normal test results, normal being high end of normal or a low end of normal, what does that really mean? So even in these conferences, I don't know whether you got an opportunity to attend a meeting on reference intervals. And nowadays, there's a lot of research going on that reference intervals needs to be customized to an individual. It varies from gender, age, and also disease. So there has to be a reference interval for healthy and a reference interval for different types of disease so that we can actually see that when we see a value, what exactly that means. So AI has a phenomenal role to play in providing that support to the clinicians when they are faced with a diagnostic decision, and typically an internist or a hospitalist who see a number of patients because their area is very broad. So they have literally figured out, what am I going to do with the patient? What is my first line of treatment? Whom do I refer this patient to? And it is extremely important at that point that if we have a clinical decision support that can help them, it'll have an immense help. So Let me go through this. So there are two opportunities here, especially on the cognitive part of the brain-to-brain loop. Especially when you are ready to order the test, we can pick the right test that will give the maximum information to be able to rule out the diagnosis, or at the end of the loop, we'll be able to interpret the results. What exactly does that mean? So that also depends upon what exactly is what you want to do. Let us say a COVID test, for example, right? So there, you really want a very high negative predictive value, almost close to 100. So if a test is negative, you should be able to confidently say that this patient is almost 100% not having COVID at the expense of a false positive. That is okay because they are going to undergo additional testing. Okay, so if you're looking at the antigen test, if it has got a high negative predictive value, then you can go through the RT-PCR, which is much more accurate. We can do that. And let us say if there is a disease which has got a treatment which is very risky and very expensive, the typical example that comes to my mind is lung nodules, right? If you have a huge lung nodules and you want to do a biopsy, it would be better if you know whether the nodule is benign or malignant, because there are instances in which patients have undergone biopsies and actually died because of the complications of the surgery. So, and then turned out to be that biopsy turned out to be benign. So, we could have left that patient alone, right? So that is where the lab diagnostics has an important role to play. CDC says 70% of the diagnosis is made based on the lab test results. And if you were here during the opening ceremony, the Ifcc chairman, Dr. Adeli, said that 90% of the data that is present in the electronic medical record is very, very objective. And majority of that is derived from the lab. So you can also see that there is a lot more effort now for integrated diagnosis, radiomics and things like that, where if the lab and imaging can combine together, work together, then they'll be able to help each other and we'll be able to achieve a very high degree of diagnostic accuracy. So there's a lot of opportunities for AI and machine learning to be able to help. So I want to show an example what we did. Okay, so way before the COVID, when the COVID started, as you all know, that hospitals are overflowing with COVID patients. The ICU is completely filled up. And physicians were really struggling to figure out what to do. And it is kind of interesting because I was very much aware of that situation where there was a scarcity of intensivists and pulmonary physicians. So they were recruiting even pediatricians to take care of COVID patients who have never done this. And then, as you know, a lot of people were put on the ventilator, which shouldn't have been done. So there's a lot of chaos and things going on during the COVID. So what we did was we were able to collect data from multiple sources. The picture that you see here is from University of La Paz Medical Center in Spain. We collected quite a lot of data from there. And in addition to that, we also collected from 27 hospitals across 3 different health systems in the US, in Houston, Atlanta, and New York, close to about 14,000 cases. And then what we did was we built an algorithm, machine learning algorithm. So basically, we had all the data, and then we also knew what really happened to the patient. Some of them were very mild diseases. They were discharged. And then some of them actually ended up in the ventilator, and a few of them had multi-organ failure, and then, unfortunately, a few of them died within 30 days of admission. So we collected over 60 different parameters, including comorbidities, vitals, and then trained a machine learning algorithm to learn, it is a supervised learning, to learn whether they can predict what the risk is going to be for a COVID patient who are all RT-PCR. RT-PCR positive patient comes to the hospital, you can run the algorithm based on the parameters. So I'm going to talk about the parameters. The main parameters, so basically we actually started with about 60 plus parameters and trained it. And we got an ROC of 78%. And then we tweaked it so that we have a very high negative predictive value. As I said, it's extremely important to have a high negative predictive value. So we were, and another thing is that 69 seems to be quite a lot of parameters. So I want to just give you a little bit more detail about the type of patients that we got. So it is listed here as sites A, B, C, and D. And at the bottom, you see the distribution of the patients. So what we did was we actually divided into a score of severity score of 0 to 4. 0 being not at all severe, they got discharged after an initial assessment. 1, they were given oxygen through a nasal cannula. And 2, they were put on the ventilator. 3, they had one of the organ failure, multi-organ failure. And the 4th, they passed away. So if you look at the distribution of patients across these 4 sites, you see there is a lot of patients with 0 and 1, very mild cases, and majority of them actually, got better after oxygen. And then only a small percentage of the people actually got into, ventilator or an organ failure, except in Site C, Atlanta. Atlanta, you see quite a lot of people who got connected to the ventilator. They had multi-organ failure, and they also passed away. And same is the case, I think if you look at the mortality, we see a high mortality in Madrid and in Atlanta. So this is actually an AOC curve for each one of these. See actually 5 lines for each of the curves. The top one, where we used all the 69 parameters and 11 comorbidities, and we were able to get an AUC of 0.78. And then we scaled it down to about 9 lab parameters, okay, which I'm going to show you in the next slides. So even with 9 parameters, we were able to get an accuracy of 0.74, which actually is pretty good because we had this available as a evaluation use only thing on the web, so that physician will be able to, or nurse will be able to enter the data into that interface and should be able to get a response back. But ideally, it needs to be integrated with the lab system so that physicians won't need to enter anything. It automatically goes from when physicians order in the lab information system, it goes into the instruments, the results get back, and the results can be sent to a model that actually does the processing and then get the results back, which can be sent back to the LIS so that physicians will be able to see that and take action. So if you look at the next slide, these are the 9 parameters that we scale down to. If you look at it, just the age and hematology, like neutrophils and lymphocyte, 2 coagulation parameters, D-dimer and INR, and immunochemistry, creatinine, LDH, high sensitivity troponin, CRP, and ferritin. And if you must have read a lot of papers, and this shows up in multiple papers being an important markers for COVID. So one of the important things, this is Dr. Munoz, who worked on this project, especially he is the lab director from La Paz University Hospital, Madrid. And he said that AI-based lab test support interpretation is extremely important. Especially in the current state, when I said, the number of lab has exploded, the physicians don't have time, they really need the support. And if we can provide as a lab, we should be able to provide a lot more than just reporting the results and the reference intervals. We should be able to give them, probabilities, what are the risk scores, and then also help them to even order additional tests so that we'll be able to improve the accuracy. Maybe a sequence of testing that'll increase the predictive value so that they'll be able to take a decision. So using probabilities, we can do pre-probability and post-probability for cascading test, and we'll be able to do a much better job. So there's a lot of opportunities for labs to be able to provide that support. And another thing Dr. Bunio said is that it's extremely important that any kind of solution has to be aligned with the workflow, because physicians are very busy and they are not going to go somewhere else into a different computer or a workstations entering stuff. So it has to be completely integrated right on their workflow so that they'll be able to take the decision when they really need it. That is also extremely important. So we should be able to give them appropriate scores and also explanations about that so that they'll be able to take the appropriate decision. So that's extremely important. So this is my favorite slide. So blood, right, which is one of the main important, raw materials for the labs, in addition to urine and other things, it never lies. So this is a book written by James Laval. And if you think about it, the blood circulates through our body about 10,000 times a day, right from the head to foot. It perfuses all the organs, get all the metabolites and things like that from the organs. So it is completely impregnated with information. So we have barely touched on the surface of this, and there's yet to be a lot of things that yet to be discovered. I read an article about the value of creatinine in determining, even in predicting early strokes or early brain injuries. So it's just amazing. Who would have thought creatinine has anything to do with the brain, right? We always associate creatinine with kidney. So there are amazing number of articles on the number of, lab markers that can give a tremendous amount of information. So what I say is that blood is actually a window to human, health and disease. Which is the way my favorite thing is that, let us say medical science is a book of like 1 million pages. We have only read the first 100 pages, okay? So if an AI algorithm can say that, okay, the patient has this problem because of these lab results, then typically the ML algorithms are kind of blamed that it cannot give the explanation, it's a black box. But that is changing, actually. But why is that? Because we cannot explain. The ML can actually look at the whole 1 million pages and give you the results, just the result. But they won't be able to give you the explanation. They'll be able to give the correlation. That is going to be present in the rest of the 9,900 or 99,900 pages that are yet to be discovered. So what I think is that the machine learning can actually point further clinical research to kind of explore the correlation so that they can actually find the reason for it. So in that sense, it is phenomenal tool that not only help us to diagnose cases and treat cases today, but also expand the clinical research in future. So I think the future is going to be extremely bright for AI and machine learning, especially with advanced processing power of computers and phenomenal amount of talented data scientists who will be able to kind of help us here. So I'm really excited about it. So this is one of our customers from lab director from Swedish Hospital. And she said, this quote is very close to us. She said, I don't know what the future will bring. And we all know about the COVID. It came out of the blue, right? And literally paralyzed the whole world for about 2 to 3 years. And she said, I'm confident that Siemens Healthineers will provide not only the innovation, but also the partnership needed to face it. So not only Siemens, every one of these companies, every one of these professors and researchers who are working around the globe is constantly trying to find things, innovate, to be able to make the healthcare, much more, effective and reducing the cost, improving the care. And in fact, preventing. I mean, we don't want patients to come in a very critical position. How can we detect diseases at a very early stage and prevent it? The couple of slides, the last slides Dr. Wheeler said, that is the kind of a vision, the future, right? I mean, in terms of all the modalities, diagnostics, imaging, advanced diagnostics, and things like that coming together, and should be able to help, physicians, in fact, a multidisciplinary team to be able to take the appropriate, intervention. So that is the vision, and I think we are definitely moving towards that. And I see some of that even in this conference, where I see a lot of imaging experts talking about integrated diagnostics. So I'm really thrilled about that. I think we are ready to take some questions. We have about 12 to 13 minutes. If you have any questions, so please, for me or Dr. Wheeler, if you can step in front of the microphone so that everybody will be able to hear the questions. So if there is no questions, if you want to kind of share your experience with AI and machine learning and want to share some of those things, definitely like to hear your opinion. And if you heard something in this conference, if you want to share something, that'll be fabulous as well in this area. So yeah. So if you want to share some of those things, yes. There is one question. If you want to introduce yourself and ask the question.
Nice to meet you, Anna Carobene from Italy. I will be the chair of the strategic conference. And I have two questions for you. The first, if the database that you use for the algorithm is available, if I can use, if it is available for the scientific community. The second, if you have published your data, and also because I have published with machine learning for the COVID, for both diagnostic and prognostic studies. And I would like also to know your opinion about the validity of the algorithm, also with other population, with the COVID positive patients now that are different from the patient that we had a couple of years ago. And if your database is only, I think so, actually I had a lot of questions for you, not just one. If is only as with the Siemens data, I think so. And if the algorithm is automatic in your instruments, because I didn't check this point. Thank you.
Thank you. A fabulous question. So I will take in sequence. Number one is the data is not publicly available. So we had some partnership with the hospitals for getting the de-identified data before we ran the algorithm. Your second question is that, yes, that work has already been published. I can definitely send you the link for that. And thirdly, there are public databases available. So you may want to, especially for research purposes, so not for commercial purposes, it's available. So I can definitely give you some of those links that you can download it. You need to sign off some agreements and things like that, just to make sure that you use it only for research purposes or test any kind of concept and things like that. So you can definitely do that. And to your final questions, if I missed, please tell me. Yes, we actually built this algorithm way back early 2021, right? At that time, there was no vaccination, there was no Delta, there was no Omicron. And then we did a couple of evaluations after the vaccination with patients with 1 dose, 2 dose, booster dose, and also with Omicron. We haven't, and also with Delta. We haven't done Omicron yet. So what we found out that, which is kind of interesting because we thought that we may have to retrain the algorithm because it's a totally different, right? But we found the accuracy is holding even with that patient. So, I was thinking, why is that, right? So maybe is that people who are taking medications and people who are also vaccinated, some of those parameters are actually going towards normalizing it. So in other words, let us say the patient is actually taking medications, the D-dimer value is probably coming down, and the algorithm actually is able to sense that and giving a lower risk score, if that makes sense. So whatever the medicine or the vaccination is actually reflecting in the parameters values, and then the algorithm is able to kind of assess the risk appropriately, which was a great surprise for us. We are continuing to do evaluation. So the whole idea is that hopefully COVID is going away and maybe it's endemic, but you can actually use this concept to be able to do any kind of, any kind of targeted diseases or any kind of, diagnosis, risks score, things like that. So did I get all your questions? Is there anything that I missed? Okay.
Did you try to evaluate your algorithm with another source of data? I mean, an external validation with the data not from Siemens?
Yeah, I mean, so we used different data from different hospital systems, right? US as well as Spain. And we evaluated with a hospital in India and also a hospital in the Middle East. So as you can see, the patient population is totally different, and it was able to give consistent results. We have only done retrospective studies. So the next thing is prospective studies, so that, and also I missed one of your questions. You asked me whether this algorithm is actually part of the instrument, right? The problem is that according to the regulation, if there is a software within the instrument, it is considered to be, software within an instrument. It needs a complete total regulations by the GDPR, by the FDA. So if it is separate from the instrument, then you could possibly get an exemption from the FDA, because it's a separate software and the physician will have the option to act on it or not act on it. So the software is not going to take the action. So in that case, it is much more safer rather than if it is in the instrument. So, so it is not in the instrument, it is totally separate, it is interfaced with the instrument. So if it is within the instrument, that is a whole different regulatory process that we have to go through. Yes. And also, I think things probably are going to change because these AI-based algorithms are coming. And even the regulatory agencies are not very familiar with this kind of regulatory things. And there are a lot of, organizations, Siemens and other companies are trying to educate even the government about this kind of software so that, they can also make sure that, because, it's a double-edged sword. If they regulate too much, then it cannot be used for good, it cannot be put for good use, right? So long as we can assure that it is not going to harm the patient, it's actually going to help the physician, then, definitely, there should be a path forward. So most of the software that you see in the market are actually calculators. So you are familiar with QSOFA scores and things like that for multi-organ failure. It is just taking multiple lab results values and calculating it, which is actually open and anybody can build it. It is not any kind of proprietary software, right? So, but I think things are coming, but the only thing that I feel the software can be approved in future is when we do multiple evaluation studies. Just to make sure that we have to present it with multiple evaluation studies, multiple population, different ethnic group, just to make sure that it works. That probably is the way to go. And GDPR, I think, is a little bit more stricter than FDA, so they have their own regulations. And I heard in Europe that even every country has got their own regulation that actually sits on the top of GDPR. So, yeah, interesting. We got to wait to see where it goes. So I have Dr. Wheeler, do you have any comments on that question from your experience?
Yeah, so I think, I guess two. My first is that right now, one of the things that we're trying to do is do a lot of things on the back end with the lab, because that's something that is not overly regulated and where we can be of help. And then the other part is just that we need to be involved in these discussions with regulators around appropriate ways to regulate this, because we have as a clinical laboratory community, we have the expertise to be able to inform this well, so that it can continue to be of use and not become so overly regulated that we can't actually get the use out of it that we should, but can still be safe.
Good point. Really good point. Thank you. We have a couple of minutes. 2 more minutes. So any more? Yes, please.
Hello, doctors. Thank you very much for the presentation. It was really impressive. I do have a question. I was very happy to see this slides. You show the brain-to-brain loop. You talk about 2 pieces. The beginning part is the test ordering. The end part is the test interpretations. And you talked a lot about what you mentioned, that the physicians during their education, they did not have enough time to spend to really understand what kind of tests to order and how to interpret it. So that was really a kind of pain point, an issue there, right? So your solution has been really tackling on how to interpret the tests by using AI, the real world data, things like that. But for the beginning of this loop, like how to support the physicians to order what tests, what are the right tests, what are your prospects, what are your visions on top of that from Siemens?
Thank you for that question. Yeah, that's a kind of a loaded question, actually. Number one is, we need to get sufficient information about the patient in terms of signs and symptoms, in terms of physical examinations, so that we can actually recommend the probable test that is going to add value, right? That is one thing. So there are a lot of databases that are already available, so we can use them, right, to be able to. And also in terms of how relevant a particular test is. And also, just Dr. Wheeler was talking about the pre-test and the post-test probabilities, right? So most of this accuracy of this test depend upon, sensitivity, specificity, like false positive, false negative, all depends upon what is the pre-test probability, or in other words, what is the prevalence rate, right? So if the prevalence rate is 50%, then any test will be 50% accurate because it is like flipping a coin, right? So if the prevalence is only 1%, then it's a difficult task. Then any test will be able to have a very high negative predictive value because 99% of the patients are not going to have the disease because prevalence is only 1%, right? So the sensitivity and specificity actually depends upon the prevalence rate, right? So to take advantage of that is cascading tests. So if we know the prevalence, and then if the test can actually increase the probability, so prevalence is 1%. So if a test can increase the probability from 1 to 5, for example, right? And then the second test will have a pre-test probability of 5. So, and then it can boost it up to 25 or 30. So right now, what we are doing is that we are looking at all the tests all together and trying to figure out whether it's normal or abnormal. But we need to have some kind of a statistics into this interpretation so that we can kind of cascade the pre-post-test probabilities depending upon what you really want to do. Do you want to increase the negative predictive value or positive predictive value, depending upon what exactly is your decision? So based on what you are tasked to do, we can actually do this, right? So the thing is, what is the pretest? What is the prevalence? Prevalence is very, very important. So, I mean, you can get the prevalence from the literature. That may be different from a particular area. We can look at the census. You can go to the local health, and there may be during like seasonality as well. During the flu season, there will be a lot more of, lung infections. So if you know that, then you'll be able to do that. So I don't know if I answer your question or not, but we will be able to take care of not only like machine learning, but also statistical models and Bayesian probabilities to be able to do a better job of helping physicians. Dr. Wheeler, do you have anything to add? Because Dr. Wheeler actually showed that during the HIV testing by cascading the probabilities, because started out, the first one was have a very high false positive rate. Then she showed how it actually enriched during a sequence of machine learning algorithms.
Yeah, so we consider that a multi-prong approach, and I will be brief here. So we are trying to be at the table more when we're looking at educating medical students and residents about what they should order. We're also trying to handle a lot of cascade testing within the laboratory. So we might call something an HIV screen and then take care of the appropriate cascading behind the scenes in the laboratory, so that we're really answering the physician's question of, does my patient have HIV, not just running an HIV antibody test. That doesn't work for all testing. And so the other piece that we're doing is being at the table for EMR discussions, where kind of care packages and standard of care packages are being created, so that when physicians are interacting with the EMR and they say, hey, I think my patient has hepatitis, and they go into the hepatitis standard of care package, there are actually all of the appropriate tests are already there, and we're not making it easy for them to find kind of the rarer tests. They would actually need to refer to a specialist to get to these more specialized tests for things that have very low prevalence. But I think that there's a lot more that we can do there, and being at the table for those EMR decisions has been really key in improving that.
Thank you, Dr. Wheeler. I think we are over the time, so if you have any more questions, please reach out to me. We can definitely discuss, and hope to see you guys next year. Thank you for attending. Thank you, Dr. Wheeler.
Thank you, everyone. Have a good day.